In this presentation, I will describe how we obtain dense 3D reconstructions from video footage monitoring conveyor systems on board fishing vessels. This research was carried out in collaboration with colleagues from Marine Scotland Science, Aberdeen, and the Centre for Environment, Fishes and Aquaculture Sciences, Lowestoft. The work was funded by the EU's Horizon 2020 programme. This presentation provides an insight into the efficacy of the approach. More details are available in an article published in IET Image Processing. This is free to download and can be accessed via the link provided on the next slide. Conveyor belts are a common feature on board UK fishing vessels and are typically used to move fish from a holding tank to an area where they are sorted, gutted and packed. Once sorted, any unwanted bycatch is usually discarded. In the last 10 to 15 years, remote electronic monitoring systems have been developed to monitor fishing effort. Remote electronic monitoring, or REM, logs a variety of signals and records video that can be used to determine the status of the vessel. Traditionally, those responsible for the management of fisheries have relied on human observers aboard vessels to verify compliance with the regulatory frameworks that govern allowable catch and discards. However, REM raises the possibility that this task could be undertaken remotely. Trials of REM video surveillance by Marine Scotland Science, undertaken over a decade ago, demonstrate that this is indeed the case, but that the human analysis of video footage is a bottleneck. The video loop we see on the right was recorded by a REM camera on board Marine Scotland's research vessel MRV Scotia. UEA has been working with Marine Scotland Science since 2013 and with CFAS since 2017 to investigate and develop computer vision systems that enable REM video footage to be analysed automatically. This work has resulted in a system called Catch Monitor. Catch Monitor is a deep neural network that has been trained to identify and count a subset of fish species that are common in UK fisheries. The system is able to segment and classify individual fish on conveyor belts and sorting tables. This represents a very challenging environment for computer vision. The training database for Catch Monitor comprises several thousand individuals labelled by marine scientists at CFAS and Marine Scotland Science. As the database evolves, the number of different species grows, and currently it includes about 30 species of fish. However, the images on the slide have been taken from an article published in 2020 and shows a confusion matrix for 8 common species. The manual task of isolating and labelling individual fish is a significant factor in improving this technology. Also, certain fish species are underrepresented in the database compared with other commercially important species, and this affects performance. We hope to address these factors by training on synthetic fish imagery. Preliminary work using generic 3D models demonstrates this yields a performance improvement. Motion stereo offers the possibility of obtaining more realistic 3D fish models for this endeavour. Here we can see a typical motion stereo 3D reconstruction. The raw video we presented previously has been transformed and the conveyor belt has been cropped. The resulting depth reconstruction is not perfect. We will discuss the reasons for this later, but first we will describe the method we use. Motion stereo is not a new concept. It was originally proposed in the 1970s, and further research was published in the 1990s. But since the 1980s, motion stereo has been lost in the large body of work on structure from motion. Motion stereo can be described as a constrained form of structure from motion, in which the subject undergoes a translation as a result of motion due to the conveyor belt. As we can see from the diagram, this results in a very similar geometry to that of conventional stereo, and so we use the standard conventional stereo pipeline to recover the 3D reconstruction. So why bother with motion stereo when conventional stereo is an easier and more established approach? The answer is that we have several hundreds of hours of legacy video footage recorded by a single REM camera, and recapturing this with a stereo camera or another depth sensor would be very expensive. Although at first sight, applying motion stereo might seem fairly straightforward, there are some particular engineering challenges presented by REM video that make the problem a little more difficult. Firstly, REM cameras run at 30 frames per second, but the systems archive video at a reduced frame rate. This introduces some sampling inconsistencies, which are important when it comes to tracking motion. Secondly, REM cameras employ CCD sensors and rolling shutters. Rolling shutters introduce motion artifacts that are difficult to correct. Fortunately, the linear conveyor motion is easy to model, and so we can correct for motion effects introduced by the rolling shutter. Thirdly, the conveyor, in this case on board Marine Scotland's research vessel MRV Scotia, is featureless, and this makes accurately tracking the conveyor motion difficult. We need to do this very accurately because the accumulated motion represents the stereo baseline. We use a Kalman filter to predict the belt position from a set of measurements taken from the belt. Finally, our stereo pipeline needs to deal with varying illumination. If you review the footage, you will see highlights and shadows on the belt. We use a convolutional neural network to match image patches and generate disparity. In order to recover real-world measurements from our camera system, we first need to calibrate it. We do this by acquiring a set of images comprising views of a checkerboard placed in different positions on the conveyor belt. Marine Scotland acquired calibration video for each fishing vessel and for their research vessel, Scotia. We use a standard camera calibration pipeline to recover the camera intrinsic matrix, the camera extrinsic matrix, and lens distortion coefficients. We also find a perspective transform that maps a manually chosen region of interest corresponding to the belt to a rectilinear space. The resulting rectified imagery forms the input to the stereo pipeline. Here we explain how we measure the stereo baseline. First, we need to identify pixels in the image that represent the conveyor belt. Since foreground pixels representing fish will suffer disparities that may compromise our motion estimates. We previously developed a mask RCNN network for our fish species classifier, so we reuse it. Next, we find scale invariant feature points that lie on the belt and match these with SIF points in the next frame. The resulting motion vectors are shown in B. The X component of measurements are assembled as a histogram, and the most frequent is chosen as the best motion estimate. These estimates drive a Kalman filter that predicts the belt motion increment between successive video frames. We continue to accumulate belt motion increments until we achieve a minimum baseline. We use the stereo pipeline proposed by Bontar and LeCun. This represents an early attempt at stereo matching using a convolutional neural network, and it outperformed all other approaches on the Middlebury benchmark in 2017. Neural networks represent the best performing approaches to stereo matching, although results from more recent networks improve on Bontar's result. The network matches patches of 11 by 11 pixels and outputs a cost disparity volume, which is subsequently processed by the stereo method pipeline proposed by Hirschmuller. The pipeline is implemented in CUDA and runs at about 5 frames per second. We reduce the search space by first translating one image by the stereo baseline. We also render the background in the left and right images with the same pseudo-random texture, as this ensures background patches match with zero disparity. It is impossible to assess the accuracy of the resulting models from the REM camera because we have no ground truth depth. So we built a rudimentary laboratory model. We fixed Heroku markers to the belt to simplify motion tracking and avoid the need for a Kalman filter. We used a budget stereo camera fixed about 750 mm above the conveyor. By constructing a highly textured calibration model and associated CAD model, we were able to compare motion stereo and conventional stereo. The results demonstrate there is little difference, but because motion stereo acquires left and right images at different times and conventional stereo acquires them simultaneously, the noise performance of conventional stereo is slightly better. Here we see two 3D reconstructions produced by motion stereo. The left uses standard resolution and the right high resolution. Part of the fish is included and cannot be reconstructed. We can now return to the video shown earlier and discuss some of the problems. Firstly, when fish are pulled from the conveyor, the motion between left and right images is corrupted, and it is no longer just due to the conveyor translation. Under these circumstances, the stereo pipeline fails and the image breaks up. Secondly, mask RCNN occasionally fails to segment smaller fish. Thirdly, sometimes the motion tracker fails to identify and match sufficient SIF points to accurately recover the baseline, and this introduces inaccuracy in the disparity map. In conclusion, we have seen that motion stereo enables generation of 3D models from legacy REM video footage. It is not without its problems. We have seen that not all frames from depth video are good quality, but there are enough to justify the endeavor. Experiments using the laboratory rig show the performance is comparable with conventional stereo. Future work will incorporate the 3D reconstructed models in the training set for our species classifier. Preliminary results using a generic roundfish depth profile suggest an improvement in performance of about 20% is possible. Thank you for your interest in our research. Catch Monitor is only one of a number of technologies developed within the SmartFish H2020 project. Details of all the research and development can be found on the SmartFish web portal. Google SmartFish for more information.